Okay, so we'll move to the second section, which is a protein. Tuesday, Dr. Kenny will give a guest lecture about the muscle protein, which is related to some amino acid knowledge. So today, we're going to do a review of overall protein and talk about amino acids and those basic knowledge. So protein, it is coming from a Greek word. It's called Protein, which means first kind. So that means first kind, important. Now obviously we know the biological function of protein is a lot. We mentioned in different classes, like it is a support function or muscle protein. It is for transport. Let's say the transmembrane protein in the cell wall, cell membrane system. Could be a hormone, which is a very small, tiny chemicals, but will play an important role. It's also could be a defense, and we say it's a protection. For example, like our antibody. We mentioned in the different classes the antibody structure. It looks like this. S double sulfide bonds. We have a H heavy chain and we have a light chain, and this is, we also call it FC section, and these here we call it a variable section, which is obviously combined with antigen, so we have an antigen antibody reaction. We also going to talk about the key thing here will be enzyme is protein. There's lots of the enzyme related to the biological function, and in the food chemistry there is a separate section talk about the enzyme. We may not have time to go over it, but I just want to tell you different food products have the enzymes. That's a major section. Some of them we will mention, like a lipase. During the lipids oxidation, we'll mention that. But they do have a certain chapter talk about enzyme. Number 3 I want to mention is what type of the protein we have. We can say there is a different category. The first category, what we can say is, we call it a simple protein. So simple protein, just give you some example. We have albumin. This is like our egg albumin. So it is difficult to get the eggs these days, so the egg albumin. This is going to be, usually we say it's dissolved in salt-free water. And the second one we call it globulin. This is the one we will go detail some of these examples. So let's say myosin, and actin cross-link relate to the food meat tenderization, and also glycine, which is in soybean protein, and beta-carboxylene, which is also related to the soybean protein. We will mention later on. So that's what I did as a research when I started to do food and microbiology research, like almost 20 years ago in China. We were focused on those soybean proteins. We also gonna have another one is glutein. So we have like glutathione in like wheat. And then the last one we could have like histone, for example. when we talk about histone, we will mention those protein is major composed by like lysine and arginine. Okay, so these are the, we call it a simple protein. Now, most of the time what we have is a conjugated protein. Conjugated protein is usually as a protein is composed by other macromolecules. So we have maybe a phosphor, we may have a phosphor protein. We may have a nuclear protein. We may have a glucoprotein that is composed with carbohydrates. And we can have a lipoprotein with lipids. And very important, in our lab, we may have a chromoprotein that is related to the color of the food products. So let's say you have a hemoglobin, and you have myoglobin sometimes. Myoglobin, hemoglobin related to the color. Okay, these are related to the color of the food products. So that's conjugated protein. We also gonna say there is a other category. It's going to be the relative protein. That is a very simple example. Is so-called tryptophan or like peptide. So in the lab, we are using a very simple dilution salt, 0.1% BPW. That B refers as a buffered. Okay, W is water, so what is P means? That means peptide. This is an enzymatic products. And from the widely different protein resource. So it's going to be the relative protein. So these are some of the category about the protein. Just like opening remarks, and we go over them. Okay, next one. We are not talking about genetics, but we will be very simple talk about how the protein will be synthesized from the genetic standpoint. Just go very quickly to review, because I think this basic knowledge we should know. So in the genetic standpoint, we talk about is a central dogma, just real quick. So we have a DNA. It's going to be going through transcription, become RNA, and RNA goes through translation. They should have amino acids at the beginning, then we'll synthesize like a protein. Okay, that's called the central dogma. So let's give you a very simple example. If I have a DNA double sequence, I have like a 3' to 5', 5' to 3'. And then you have a double bonds. Let's say I have a coding which is A, T, G, A, C, G. Now, based on gene corresponding policy, we have a T, A, C, and TGC. Okay, so we call this is a coding band, and this is a template band. And then we will synthesize is mRNA. So mRNA will be right here. Now be careful about the 5' to 3'. So that's mRNA. And mRNA should be the sequencing is exactly the same as a coding band. The only difference is because it's not DNA, so we have AUG, which is T replaced by U, and then become ACG. Now once you have a mRNA, it's gonna have a tRNA comes out. And tRNA also corresponding to that mRNA. That's 5' to 3'. So since we talk about the chemistry, so what are the structure of the tRNA, how they connected? Later on, we can talk about that. For example, here we could become is UAC, okay, this is becomes a UGC. So how they connected with each other? So let's say we had a carbon right here. Okay, that's a 5-carbon sugar, and on the 1' side we had a base. We connected to the base. A base is AUCG, and then we go to the 2' of the carbon. We have HOH because it's a ribose, so it's H, and then we have H and OH. That's 3', and this one is important from when you synthesize the amino acids. Okay, now you go here, you're gonna have a H, then you have a CH2. When you go here, you have O, that's a phosphate. That's always important. So you go O here, then you connect it here. You have a C. Then what happened is this is a magic stuff we are coming, is a tRNA. So tRNA looks like this. If you can draw, very fun to draw it. Looks like that. And this is a 5', and that is 3'. So that's tRNA. And tRNA, from chemistry standpoint, we call it a clove leaf configuration. But very important is here at the end, it's carry-on. Here will be UAC, which is we call it anti-codon. And then you have these arms, you have these like a T theta C arm. So you have a variable arm, and then you also have a D arm. So that's a structure, very interesting. And how do we know it's a tRNA? When you do the analysis of those gene base, you see those wheeled gene base like T theta C, that could be the tRNA. Now, related to the tRNA carry-on amino acids, where we should pay much attention is this 3'. So let's say I have an amino acid is coming. We're going to go to the structure later on. I have an amino acid coming. I have this guy coming. Okay, I goes here. I goes R1, and I goes H, and I go to NH2. What happened is there is a reaction happened here is lose 1 molecule of water. So they connected. So how they connected? You can take out of these here, because water is gone, and this will become the bonds like that. Okay? And then become long and long. Now, then you go to talk about genetics. This happens is a tRNA will go to a ribosome, and then you have a 50 S subunits, you have a 30 S subunits. When this tRNA come in, and what you should have is recognize the AGG sequencing, and we call it SD Shai-Diagonal sequence. Now there's a lot of things we talk about genetics or microbiology class, we don't repeat it. So that's what it was a tRNA. And then the enzyme related to tRNA is always we call it we call it a tRNA transference. tRNA transference. And we should always remember the first amino acids, when they start to do, the codon is AUG, that represents a very important amino acids. What is that? That is called methionine. We'll get to that detail. Okay? That's methionine. In the bacteria, it's called formyl methionine. It's a little bit modified that. Okay. Now, very quick, we also know we had a 3 codon system for amino acid. And we have a number 1, number 2, and number 3 box. Since we have A, U, C, G, so the box first have a 4 possibility. So box number 2 have a 4 possibility. Number 3 have a 4 possibility. So total is 64. However, we are 61 codon for 20 amino acids because there is a 3 stop codon, and which is not for any amino acids, not translate to any amino acids, which is UAA, UGA, and UAG. Okay, so and talking about mutation, the number 3 position is flexible. That's why one amino acid has multiple different codon, and we call it a wobbling effect. So this is very quick. We spend basically 5 minutes. We go over what the genetics we should talk about is how the amino acids comes out. Now later on they do lots of the modifications and they generate the protein. And they first have peptides. Okay, so now next one we're going to talk about amino acids. We're going to go over all these amino acids details. So that's number 5 is amino acids. Okay, so the basic structure of the amino acids, everybody knows we have NH2. There is a carb C on the center, and we have H, and we have the R, and we have COOH. This C, we call it a chiral C. It's in the middle center. Because of the chiral C, so we had L amino acids, and we have D amino acids. Now, we talk about 20 amino acids. Basically, these are L amino acids, because that's the only one existing in nature environments. And D amino acids, lots of the time, is we call it isomerization, which is chemical synthesized in the lab. Now, the angle here is not really straight. There is a dimension structure which is an angle for that. So here you're gonna see like could it be P SI pizza angle, and here you have an angle we call it a theta angle. So it's not straight. Now when we got to here, the more important is these 2 ends. These 2 ends, you have a COOH, that is C, double bonds O, OH, and this is called carboxy end. And then you have NH2, this is called amino group. And we know that basically the amino acids is not neutral. They are charged. So you can write it here, the positive, right here is negative. This is what we are going to get to later on, okay? So this is amino acids, the basic structure. Amino acids, we talk about like 20 amino acids. So 20 amino acids, basically the difference is the R. So we're going to go over the R, what are these side chains, or the difference. Now we can do a categorized before we go those details. So what's a categorized? We talk about the polar, non-polar, so the amino acids is the same thing. It could be non-polar. Now non-polar amino acids, basically they cannot dissolve into water. So we call it hydrophobic. And then we also have some of the polar amino acids. Those are, we usually say, hydrophobic hydrophilic. And the hydrophilic, that could have two different categories, which is no charge. They dissolve in the water, but they no charge, or they could be charged. And when they charged, they also going to have two different categories that comes out. There could be acid. It's quotation mark acid. It means negatively charged. It also could be basic, and that is positively charged. Okay, so these are the basic information related to amino acids. So then we're going to go over to see the all-side chain, what are they looks like. I used to can remember them very well because I have a busy in the morning. I'm revising manuscript, so I may forgot some, so we'll see. The first category is non-polar. Non-polar, we say obviously is no charge. Okay, so the first one, let's go this. First one is H. This is glycine. Okay, we talk about amino acids is a chiral C. But if this is glycine, that is an exception. That's not a chiral C. There is a no L and D because it's both are H. The second one, CH3. It is alanine. Then go a little bit complicated. We got CH, we have CH3, then go CH3. It is called valine. Valine, when I do, I usually using a red color. I don't know whether I have this. I usually using a red color to highlight because this is the first essential amino acids. Okay, then we go next one. We have CH2, CH, then you go CH3, then you go CH3, that becomes leucine. And leucine is the same thing as valine, it's an essential amino acids. So I usually mark it red. And then you have a leucine, it's very simple. They have isomerized, which is called isoleucine. So we go here, CH3, so we go CH2, and then we go CH3. That is isoleucine. Okay, then the next one, a little bit tricky. The next one, we have to draw the whole thing. Because it is a very exception, it is a so-called a circle. So you have to draw CH2, CH2, and then you go CH2, and you go here. That is all together, so we call this as a proline. Okay, this is the one, we got the first one. Then we started to see something a little bit difficult structure. Why it is difficult? There's a phenyl group gonna come in. So you have alanine. I'm gonna add a phenyl group. So I'm gonna add a phenyl group there, like this. Sometimes you just draw a circle, then you become a CH2. What is this called? Phenylalanine. Okay, the next one is more difficult. That one, lots of people don't like it, because I don't like it too, because it's too complicated. Looks like. So kind of like this. Go here, 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 NH. Okay, then you go here, CH2. What is this guy called? Tryptophan. Tryptophan we will mention, okay? A little bit more, tryptophan. Okay, once you have a tryptophan, then the next one, by the way, phenylalanine and tryptophan are also essential amino acids, so we mark it red. Then the next one is started to have a debate. So I look at the textbook. Lots of the textbook put this guy as no charge. Some of them, they put it as a charge, which is cysteine. So I do put this one as no charge. but you are microbiology, maybe your biochemistry class, maybe put this as a charge. Cysteine. Cysteine is important. They start to show you the S.
Does phenylalanine have an oxygen in the center?
No, I just draw like that because there's two different expressions.
Oh, okay.
That's why I will
that
draw a
was.
white one.
Okay.
You can take it out.
Oh.
Okay, do you know that? Let's go this. That's why we want to go chemistry. Phenol group, is that right? Could be like this.
Oh.
Is that right? Is that phenol group could be like that? Is this? So, I want you to know, that's why I draw like a white one. Is that good?
Yeah.
Okay. That's why I said for chemistry we have to go over it, okay? That's for the chemistry class. Okay, cysteine. Cysteine we have start to have like a sulfide group. Okay, next one we already mentioned. That's a very important one. CH3, S, CH2, CH2. That's methionine. Okay, methionine, always important. That's the first one. We do translation transcription. And that's a S. Okay, this is obviously is essential amino acids. Okay, when we get down here, this first group is done. No polar, no charge. Then the second group we start to have is polar, but no charge. So what this polar means? It is starting to become a hydrophilic. Which means they can dissolve in the water. Okay, when these things happen, obviously, this guy gonna show up. Hydroxy bonds gonna show up. So, we started to do is the second group, is CH2OH. That is serine. And then we have CH3OH. That is called serine. Okay? I don't know the reason, but serine is essential amino acids. Okay, then we go to interesting one. We had CH. This is CH. This is goes to O. Then you have NH2. This is called asparagine. You know, we want to make sure CH2S. That's why I said I forgot about it. Sometimes CH goes C goes here, then NH2. That's asparagine. And then you're gonna add one more there. So we're gonna add CH2, CH2. You have a C. This guy called glutamate, or glutamine sometimes, because of glutamine. Okay, last one. Don't forget this group has one, also has a phenol shows up. Go here, go OH, CH2, that's called tyrosine. Okay, when this group is done, we move on to the last group, which is polar and charged. That's very easy for the first two. We have asparagine. Then very natural, we have CH2, C. That's called aspartic acid. And then you have a glutamate. You have a glutamate right here. It's very easy, the same thing. Just add a one CH. So CH2, CH2, go C. This is called glutamic acid. Now, these are negatively charged. That's why these two, we call it acid. Since we are talking about acid, then we're going to talk about is a basic. So the last couple, we talk about the basic. Okay, what are the basic one? The first one, this draw always difficult. NH, plus, go here, go here, and then CH2. Histidine. Then the next one, we will mention later on. Very long CH. CH2, CH2, CH2. See, not even enough space. CH2, then go NH3 plus. Lysine. Now, this is an essential amino acid. Last one, arginine. Arginine very similar to lysine, little bit difference. So CH2, 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 we go C, then NH2, double bonds, NH2 is plus. So that's arginine. That's also an essential amino acids. Okay, so this is done. And these we call basic. That's positively charged. Okay, now we want to go over something. Other classes we mentioned. Phenylalanine. Phenylalanine, we in the microbiology class, we talk about the deaminase test. Deaminase test, called the phenylalanine deaminase test, because a bacteria called deaminase generates a side product which is called phenylpyruvate. Same as a pyruvic acids can go through citric acid cycles, they can survive, which means they can use other energy resource for the bacteria to survive. And how do we know it is positive? We add a ferric chloride, which is showing dark green color. The second one I want to talk is the tryptophan. The tryptophan is a very interesting one. If we cut from here, this guy is called Indo. Indo is a very simple test for E. coli. Regardless of E. coli 0157H7, non-0157 Shigella toxin E. coli or generic E. coli, it is always tested positive. The reagent called COVAC. Why I want to mention this is Dr. J and Dr. Guggenson worked a little bit a couple of years ago for rapid testing using GCMS to see the E. coli, which is to see whether they can using Indo test to simply to testing E. coli in the food samples using GCMS. So that's why I want to mention, okay? Methionine cysteine. That's not too much. Methionine, we already mentioned the first one. Okay. Serine, serine, asparagine, glutamine. Glutamine, do you know salt, glutamine, sodium glutamate? What is that one? That one can using a powder put in the soup and will make it very tasty, delicious. I always say very tasty, but you cannot eat that too much. That's called sodium glutamate. Aspartic acid, glutamic acid, not too much. Here is there is a joke we could talk. I talk in the other classes. So when I was students back in the 20 years ago, the joke comes from when they're teaching the amino acids in the 1990s. So by the time, we don't talk too much about the minority or DEI or those issues. So we see those young ladies or millennials working in the campus and wear those long boots, but they doesn't pay much attention to their study. So they say this is called her leggers or basic. So this is like a joke. That's why I used to remember this. So histidine, that's her, H is histidine, leggers, which means lysine, R is arginine, and those are basic, which is positively charged. Okay, so that's the way we go ahead to remember and memorize all these. Now, there's one thing we want to mention. If you look at your new textbook, or the new biochemistry textbooks, they already start talk about, we have 21 amino acids instead of we talk about 20. So what are they? What's the last one? CH, SEH. Selenon, cysteine. That's they call the 21 amino acids. I think the new textbook already showed this up. Okay. Next one, I left the part. We'll remove this joke. So take this one off. We want to mention a very important. Just review our important characteristics of amino acids. We call acid and base amino acids characteristics. So we go back to talk about the structure. We're already going through the R side chain. Okay, we have NH, I'm gonna have to write in the middle. We have NH2. We have C, we have R, H, and COOH. This looks like the structure like this. But in our normal life, it's not. This is we write NH3, positively charged. And this is we write. Lots of the time we took this out, become negatively charged. So what is this one we call? This is basically at pH around 6 or 7 neutral status. The amino acids present dipolar ions. It could be positively charged or negatively charged on the two ends, which is we call alpha amino group and alpha carboxy group is charged. So we give them a name called the Zwitter ions, or we say Zwitter status. Okay, since we have this very tricky but interesting status, there is two directions of the biochemical reaction could happen. Go this direction if we add a proton. So we call it protonated. Protonated. If the protonated, what will happen? It will have NH3 plus COH, and the proton goes here, so becomes COOH. That become a positively charged. We have a pKa1 comes out. Now it can do proton, but also it can do what? D-proton. Okay, once to D-proton, what happened? Go a different direction. That's called D-protonated. When the D-protonated, it's gonna go. So what it gonna looks like? Will become NH2COH, and this becomes COOH, and COO negative. Okay, that is have a pKa2. So this comes out with something. What is pKa1 means? This is pH at which the dissociated dissociated carboxy group the concentration equals undissociated carboxyl group. That's called pK1. Okay, then what we have is pK2. What's a pK2? Is pH at which the charged amino group equals non-charged amino group. That's called pK2. And you don't forget, there is another one tricky, is the outside chain. So that's the outside chain. We just gave it pK3. Then this comes out with the next question, the very important one. We talk about at pH 6 or 7, the amino acids present as a Zweiter status. It could mildly carry on positive or negative, but there is a point at a certain pH. The Zweiter ions are neutral, which is no charge. This status, this pH value, we call isoelectric point, acronym PI. This is very important. Why? Because back in early 2000, Dr. J and Dr. Matak has been published about like maybe 30 or 50 papers regarding using isoelectric point technology to see the remodeling of the products, reprocessing of the products, related to the food safety, microbiology, and also the food quality. So that's why I want to introduce here, I will ask Dr. Matak, do a guest speaker almost at the end of the semester. We'll talk about application of the isoelectric point technology to see what they did in the past 20 years, most recent 10 years for these the results. Okay, so that's a very interesting topic. Okay, we had pK1, pK2, pK3. This comes out with the next question: how to get PI? Now this is go back to chemistry, biochemistry. Everybody knows there is a very important and typical curve comes out. What is that typical curve comes out? Y-axis. We have to separate them. Okay, go two different directions. Go this direction, goes here, 0.1, 0.2, 0.3, 0.9. Go to the top, we have 0.1, 0.2, goes 0.9. This one we usually draw a little bit longer. At the bottom we say it's HCl, equivalent as HCl. We use hypochlorite gases, that's called HCl. The top is Na, sodium hydroxide. This is called equivalent. And then you have a curve comes out. This is pH value. Is that right? The pH value, we go 0, we go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, blah, 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 goes 14. The curve is interesting, like this. Here, represents pKa value. Here, represents pKa2 value. And right in the middle, this is PI value. That's what we need to looking for. Okay? Now, how are we going to do the calculation of the PI value? It depends different amino acids, different all side chain. That's why we want to go over those first. Okay, so how are we going to do? It depends. There's 3 different categories. I'm going to draw on the top. Okay, category number 1. if non-polar, no charge, what's the pi value will be? That is simply equals pKa1 and pKa2 divided by 2. This is the first category. Okay, on the second category, if it is an acid which is negatively charged amino acids, what's the pi value will be? That's very easy, but it's related to the side chain. That will be pKa1 and pKa3, which is a side chain pK value divided by 2. Now, very normal, we go to third category. If it is a basic amino acid, like histidine, arginine, and lysine, what's the pi value will be? That will be pKa2 and pKa3, then you divide it by 2. So this gives you some of the quiz. We could certainly do some of the quiz. I remember most of the stuff. I don't remember pK of these things, so I have to go. If it's alanine, we can do a practice. If it's alanine, we have aspartic acids. And we had, that's the last one is arginine. Arginine. And then you have a different value. For example, this is a pKa value. That represents OH. And we have a pKa2, that's NH3. And then we have a pKa3, which is side chain R. So we can do a mass. We can do a mass. Okay, that's we'll be doing 2.34. That's 9.69. They don't have it. Next one 1.88. 9.60. 3.65. Last one 2.17.04.